Our speaker today is Rob Turesky. Rob is a professor in the Department of Medicinal Chemistry and the Masonic Chair in Cancer Causation. Rob got his Bachelor of Science degree at UMass Amherst, followed by a Ph.D. in Food and Nutrition Sciences at MIT. And then he had a bit of a nomadic existence, as he was describing earlier to me, where he worked for Nestle in Lausanne, Switzerland for about 15 years, then worked for the FDA for a while before becoming an associate professor at New York State University in the Albany area in upstate New York. And then he joined the faculty here in the Masonic Cancer Center about 10 years ago or so. And Rob's work is really important because I think the most profound impact that researchers have had on cancer is in cancer prevention. But to prevent cancer, you have to know what to avoid. And Rob's work has identified a number of these agents in a really elegant series of studies that he'll hit on today and describe it to us. So thanks very much, Rob.
Well, thanks, Chris, for the very nice introduction. So my talk will be on some of the work we've been doing on cooked meat and prostate cancer. And I'm going to just kind of look at the screen because I'm going to have a kink in my neck if I keep on looking at the screen. So we're continuously exposed to potentially hazardous chemicals in the environment through air pollution, cigarette smoke, moldy foods like corn contain mycotoxins like aflatoxin, or homeopathic medicines, unfortunately, which are viewed as safe, are not necessarily safe. They can contain various types of carcinogens, renal carcinogens, bladder carcinogens, including aristolochic acid. And one of my favorite topics is cooked meats, where many potential cancer-causing agents can occur at generally, fortunately, low levels, but over time could be hazardous and risk factor for cancer. So for many years, epidemiology was very observational. Epidemiologists would look at lifestyle factors and putative exposures and look at disease outcomes and risks such as cancer. However, over the past 20 years, with the advent of analytical mass spectrometry techniques and genomics, we can advance and better identify some of the hazardous chemicals in the diet environment and their role in cancer. So, for instance, we can now readily measure carcinogens in tobacco smoke, or example, grilled meats, and get an idea about exposures to potentially hazardous chemicals. But that gives an idea about exposure. It doesn't tell you about bioavailability. The compounds that we ingest, they need to become bioavailable and get absorbed through the GI tract. And so we can measure the bioavailable dose, for instance, by measuring the carcinogens or their metabolites in blood, urine. Some of them will accumulate in fingernails or in hair. But again, that's the available dose. What we really want to know is, for most carcinogens, they have to be metabolized by enzymes in our body. And so what we're really interested in is the biologically effective dose, the reactive intermediates that can damage DNA or proteins, for example. And then we can then try, in the case of cancer-causing agents, try to link certain DNA addicts with mutational spectra in people with cancer, and try to see whether we can correlate the mutational spectra in cancer driver genes with specific DNA addicts to give us a more firmer understanding of certain chemicals that are contributing to cancer risk. So my laboratory has several different research projects. We're funded to look at the role of tobacco smoke in bladder cancer risk. As you all know, Steve Hecht's work here, tobacco smoke is clearly a major risk factor for lung cancer, but it's also a very strong risk factor for bladder cancer. We also have research funded to examine the health and safety of herbal medicines and their role in kidney disease and kidney cancer. And also we're looking at anthracycline derivatives and novel mechanisms of their cytotoxic action in breast cancer. And again, the topic today that I will present will be on cooked meat and prostate cancer. So how do we measure chemicals and biomarkers in human studies? Well, we do it by mass spectrometry. The mass spectrometer, it's our eyes. It allows us to see things. Without mass spectrometry, we could not do our research. And these are very expensive instruments. This instrument here goes for close to $1 million from Thermo Fisher. I should probably buy more thermal stock. That's actually done very well, right, Pete? And I would like to acknowledge the generous support of the Minnesota Masonic charities who have invested in this equipment in my laboratory. We have to use it 24/7. It's an invaluable instrument for our research. So, as I said, we work on chemicals that are damaging DNA and protein. We get biopsy specimens from bladder, kidney, lung, intestinal tract, bladder. We look at specimens or chemicals in blood, accumulation in hair, and also exfoliated urinary cells for looking at DNA damage of the bladder. And so the people in my laboratory have to be really good analytical chemists. They need to know mass spectrometry to learn it very quickly. Most of the work we're doing is at trace analysis levels. We need to be able to do absolute quantification, and with the mass spectrometers, we can also unambiguously, in many cases, structurally identify the biomarker or the carcinogen in question at parts per billion or trillion levels. So this is an example. This is a classical study that was done by Colonel at the Cancer Center in Hawaii a number of years ago, where he looked at a group of Japanese immigrants who moved to the United States. And it's well known for many years that Japanese have very high incidences of stomach cancer in both men and in women. And the risk for this disease is thought to be due to salted fish, pickles, and other salted vegetables, which generate N-nitrosyl compounds, nitrosamines, which are well known to be gastric carcinogens in animal models. And what you can see is the parents who lived in Japan, they had very high incidences of stomach cancer, both for females and males. And as they moved to the United States, their first generation offspring and their second generation offspring, there was a decrease in the incidence of cancer approaching that of Caucasians in the United States. In contrast, breast cancer for women in Japan is quite low compared to the United States, but you have the opposite effect here. As they migrated, their offspring migrated in the second generation, the increase for breast cancer is going up and it's approaching that of Caucasians in the US. So this data strongly suggests that lifestyle factors or chemicals in the diet or in the environment are responsible for this changes in disease risk. And I might add that probably changing dietary habits is probably one of the most effective methods of cancer chemo prevention. And there are a number of other examples of this on migrant studies with different target sites, including prostate. So let me talk about the International Agency for Research on Cancer for a quick moment. It's part of the World Health Organization. It was established in 1969. And every year since then, they usually have 2 meetings per year where they evaluate various chemicals or lifestyle factors, tobacco smoking, use of hair dyes, and use of portable telephones on cancer risk. And they recruit epidemiologists and basic researchers such as myself or Steve Hecht, for example, to critically evaluate the mechanistic studies and the epidemiology on roles of certain exposures or lifestyle factors in cancer risk. And so I was part of the monograph that was done, about 7 years ago already, 2015, on red and processed meats. And this summary of their data was published in the Lancet Oncology in that year. And as I said, I was part of the working group for this evaluation. And what the conclusions were was that consumption of processed meat was clearly carcinogenic to humans. Red meat was classified as group 2A, which is just one notch below, as probably carcinogenic to humans. And interestingly, red meat was also positively associated with pancreatic and prostate cancer, which is what we have been concentrating on in the last 4 or 5 years. So eating well-done meats, does it contribute to prostate cancer risk? Well, epidemiology has been somewhat mixed, but recent years, there's been a trend with well-done cooked meats having an elevated risk for prostate cancer compared to eating meats that are cooked not so well done. And I don't know if this is a good analogy or not. Steve may object, I don't know, but most tobacco, except for nitroso compounds, the carcinogens form during the high temperature combustion of tobacco. It's not quite the same, but it's similar. So high temperatures, what's producing, forming, creating these carcinogens in tobacco smoke. Same analogy, it's not the same compounds, but high temperature cooking or grilling of meats also produces potential cancer causing agents. So prostate cancer is the second leading cause of cancer-related death for men in the US. Some of the questions that we are asking is what chemicals in cooked meat may cause DNA damage to the prostate and contribute to prostate cancer? And does eating a lot of well-done cooked meat, does it explain high prostate cancer risk? So epidemiology, I've just stated this, about the high incidence and second leading cause of death for cancers for men in the US. What about the etiology? Well, risk factor, age 65 or older is one of the risks. And unfortunately, yikes for me, I'm now in that age bracket. Ethnicity is a risk factor. African Americans have about a 2-time incidence, higher incidence of prostate cancer and mortality. There is some link to history, family history and genetics. The data on the environment and pesticides and carcinogens has been very inconsistent. It's certainly not at all ironclad. But I would like to talk about some of our work on red meat today. And of course, other factors such as chronic inflammation, oxidative stress contribute to the disease risk. So these are just a few examples. This is some work that was done by Poonen et al. At Case Western. And their conclusions were that well-done grilled or barbecued meat containing carcinogens, i.e. heterocyclic aromatic amines that I will discuss, could increase the risk for aggressive prostate cancer. In that study, they reported that meat cooked not well done was not a risk. Similarly, work by Sinha and colleagues reported that there was high temperature cooked meats was, what did I do with my mouse? Ugh. I can't move my mouse. Star. Oh, there it goes. Was associated with aggressive prostate cancer risk in African American men. And then a follow-up study reported that well-done cooked meat consumption with African American men ate the highest amounts of well-done cooked meats had elevated levels of the prostate-specific antigen, which is a marker for prostate cancer risk. And so this data does support a role for prostate cancer and, excuse me, a role for cooked meat in prostate cancer risk. But I must confide that I have cherry picked these studies. There are other studies that do not reach the same conclusions about risk. So what we have proposed to do for NIH is to develop biomarkers of chemical exposures and DNA damage to chemicals in cooked meat that may help us understand their contribution to genetic damage, i.e. DNA addicts, and prostate cancer risk. So I've been working on a chemical called PHIP. It's a heterocyclic aromatic amine that's formed in well-done cooked meats. It's actually been classified as a group 2B carcinogen. My opinion is that it will probably be elevated to a higher classification. The question comes down about the question of dose. PHIP, it's the only mutagen formed in cooked meats that is known to induce prostate cancer in experimental rodent models, which is quite important. And we believe its carcinogenic effects are driven by its capacity to form mutation-prone DNA addicts that can induce mutations. So the question is, does cooked meat containing FIP contribute to human prostate cancer? So this is barbecue 101, all right? I'm actually a very popular guy during the summer months. I get inundated with all these requests from news media journals about cooking meat and so forth. So I thought I would share a couple of slides that I've shared with them. So here's a couple of nice, beautiful sirloin steaks on charcoal grill. And what I would tell you is it might look nice and enticing, but don't cook your meats that well done. All right, that's like this. And what you really should be doing is cooking your meats more like this. Okay, the levels of FIP and other carcinogens would be at least tenfold lower. Okay, so if you don't understand anything that I'm talking about today, don't burn the meat. All right, got it? All right. And similarly, I don't mean to be a killjoy, but bacon has this carcinogen too. And so if you enjoy bacon, eat it like this, that's still somewhat moist and not crispy, like super crispy like this, or like this. Again, the levels of these compounds, they're gonna zoom up as you really eat meat that's well done, or bacon that's really crispy where the moisture content's been dried out. So that's important information. Okay. So okay, FIP in itself, like many pro-carcinogens, requires metabolism in our bodies to become genotoxic or potentially carcinogenic. So FIP will get absorbed in the GI tract, and essentially it has two pathways: bioactivation pathway to a reactive intermediate that can damage protein and DNA, or detoxification routes, which then get eliminated in urine or feces. So reactive metabolite can form DNA addicts. It can also bind to proteins and cause toxicity. Fortunately, we have DNA repair enzymes in our cells that can repair most DNA lesions. However, some of them can escape repair, and during cell division could either induce death, or if it's in a critical cancer driver gene during cell replication, it could misreplicate and result in a mutation that ultimately can lead to a cancerous cell. This is a little bit of an oversimplification, but this is the general scheme for this. So we have looked at several different small cohorts and developed biomarkers. And so we had individuals eating well-done cooked meats, mostly hamburgers. And we have characterized a number of different biomarkers in our cohorts. And we have established a method. FIP, actually, a small amount of it, the unmetabolized dose through first pass metabolism will be systematically go through all different organs within the body, including the hair shaft. And FIP has a very high affinity for melanin or other pigments in the hair shaft. As the hair follicle grows out, this carcinogen will reside in the cortex. In contrast, many of the other heterocyclic amines that we have analyzed have much lower affinity. FIP will form an addict with albumin that I'll talk about. We have looked at urinary metabolites, and we have looked at DNA damage, and I'll discuss that as well. So before we did these studies, actually, I had a collaboration here with Jaime Mondiano. It's already 10 years ago, where we had a pilot study, and we looked at meat eaters, and they were all positive for FIP in their hair, while vegetarians or self-reported vegetarians were not, which makes sense because this carcinogen only forms in cooked meats, poultry, and fish. So if someone's a strict vegan, they're not going to have this compound in their hair unless they're lying and they're a closet meat eater. Initially, we didn't have vegetarians, however, and so I used my dogs as I thought would be a negative control, and because this is Moses. He was a beautiful dog. I miss him terribly. Moses had a lot of FIP in his fur when he was a puppy, which really upset me. I made my lab tech repeat this 3 times because I didn't believe her data. And she still got the same data, so I did it myself. I got the same results. It's holy shit, what's he eating? And as an adult, his levels went down. But when you think about it, they have fur all over their bodies. So per surface area, I mean, they got tons of this carcinogen in their fur. I mean, I should have been wearing gloves when I worked up his fur. Another interesting thing that we saw though is that all the carcinogen resided in his black fur. There was almost nothing in his white fur. And so this proves some of the epidemiology that been reported. So, well, it seems that people that are gray-haired or light hair have less FIP in their hair than individuals who are dark hair. And so Moses, he actually proved that. I've been trying to get funded for 10 years to do more work on canine cancer. So when you think about it, these dogs, most of them have very regimented diets. They're eating the same stuff, morning, lunch, and evening, 7 days a week for their entire lifetime. I reached out to various pet food companies, including Purina, which is owned by my former company, Nestle, and they just, they should just shut me down. They didn't want to hear about this. It's one of the reasons why I left Nestle in the first place. But all right, anyways, I'm sorry, I shouldn't talk about that. But they just weren't interested in trying to improve their food product, and I find that disappointing. I'm still trying to get funded. All right, so let's talk. We did a semi-controlled feeding study with my collaborator, Loic Lamarchand at the University of Hawaii. And what we did is we had about 40 healthy young adults, and we had a 3-week pre-feeding study phase where they were on a free choice diet. We asked them to try to refrain from eating well-done cooked meats, barbecued meats. And then we had a 4-week period where they had 5 days a week for lunch. They would have a very well done cooked beef hamburger or steak or something. And then we had a 4-week washout period. And so we drew blood 2 time points during the pre-feeding, the middle of the feeding phase, and at the end of the feeding phase, and then 2 washout periods. Similarly, we collect urine at the same time, and we collected newly grown hair from the back of the earlobe, which more or less, because of the kinetics we've done, represents exposure from the last 3 months. And so we had analysis here during the pre-feeding stage, then at the end of the feeding stage, and also here post-feeding. And actually it turned out that we had to go longer than this thing because the growth rate of the hair, there's still very high levels of FIP in the hair at that point. So we did a subsequent study and said we really need about almost 3 months for the carcinogen to be cleared from those hair. So this is the analytical method we set up. We don't need a lot of hair. I mean, really, we only need 20, 30 milligrams. It's small snippets. And this is the dose response study that we saw at the University of Hawaii with their volunteers. These individuals ate different amounts of FIP. We had a measure in their grilled meats over a period of 4 weeks where we then measured their hair. And we normalized this for melanin content because, again, melanin is a variable in the levels the FIP that may accrue in their hair. And we get a relatively decent dose response. We think this is a much better gauge and estimate of intake than the food frequency questionnaire. The levels of FIP can vary by over 100-fold in cooked meats. And so that's not a very accurate, the question is not, in my opinion, is not a very accurate way of assessing this. So we get a nice dose response. We can estimate, we use this as an estimate for our future studies and what we think daily intake was. So FIP in hair is a long-lived biomarker that can be used to assess exposure to this procarcinogen, but it's the unmetabolized dose, and it doesn't provide an estimate of the biologically effective reactive intermediate that causes DNA damage. So we set up another biomarker, which is non-invasive. It's more invasive than taking snippets of hair, but we can take a small amount of somebody's blood. And there's data in the literature that showed that a wide range of different types of chemicals in the environment can react with the nucleophilic cysteine 34 amino acid residue in serum albumin. And we showed actually that FIP, or its metabolite, also binds to this amino acid in albumin. So again, FIP most of it gets metabolized in the liver to form the hydroxylamine. That's the genotoxic metabolite. We have glucuronosyl transferases, which conjugate this metabolite to stable detoxification products that get eliminated in urine or feces. However, a portion of this dose or metabolite is going to get bioactivated by sulfur-transferases or acetyl transferases to produce reactive intermediates that will undergo heterolytic cleavage to generate the nitrinium ion that can covalently act to DNA or albumin. So this is the chemistry which happens in vitro and in vivo. The hydroxylamine actually serves as a reducing agent and can oxidize the nitroso metabolite. This is the species that binds, covalently acts to the cysteine 34, and the mercaptol rearranges to the sulfonamide. And we've characterized this in vitro. This is the major site of binding of N-nitroxy-FIP, or the nitroso, to albumin. We've looked at the pentapeptide LQQ-CPF, which is at the cysteine 34. We've done MS to the seconds and MS to the thirds to get the YB ion series to prove that this is indeed the site of adduction. And this adduct, you can isolate albumin from blood. And if you treat it with acid, mild acid, you can hydrolyze this linkage to generate FIP and the cysteine sulfonic acid, and then you can isolate the FIP from the plasma, and you can do LC mass spec to measure for it. And we've inferred, based upon our other data, that this acid label adduct, we believe, is almost exclusively attributed to this sulfonamide adduct. So again, part of that controlled feeding study done with the University of Hawaii, we looked at the levels of acid label FIP addicts in albumin at days 14 and 21 of the pre-feeding. Except for one individual, all of them were low. Day 35, 2 weeks into the study, we didn't see much, but at day 49, we get a big jump up in the level of the adduct. It's ostensibly it's accumulating over time, we thought. And we have the same, we did a second group and we have the same profile. And then this is where they ceased eating at day 49. And what you can see 2 weeks later is actually more than 95% of the adduct it's gone. Unfortunately for me, because I would have a lot more funding if this was a stable adduct, this adduct is not stable. It has a half-life of well less than 72 hours. And I don't believe it can be used to biomonitor subjects on a free choice diet. It's just the rates of its hydrolysis are too much. But this data does prove that FIP gets bioactivated in humans. Interestingly, there's a 220-fold range of adduct levels, which is very interesting. It is not, however, it does not appear to be associated with CYP1A2 activity, the enzyme involved in FIP bioactivation. The level of CYP functional activity, it's bouncing up and down all over the place during the 4-week feeding study. The expression is not stable. And as I said, there's about a 200-fold range. We need about a 0.5 ml of plasma to measure this adduct. And unfortunately for me, the half-life is very short, and it's not going to be easily used in free choice diets. The urinary metabolites we can readily measure within 24 to 48 hours before they're gone. Major activation pathway is through N-oxidation to produce the genotoxic metabolite. Fortunately, we have glucuronosyl transferase enzymes that extensively detoxify it. And FIP in hair represents the long-lived biomarker of this carcinogen thus far. There's no other biomarker that can be used to my knowledge. So the next question we ask, can any of these biomarkers be employed in cancer risk assessment? So I'm sorry, this is kind of a busy table. This is a study that we did with Chris Waite and Perry Murigan, the Department of Urology here, who were treating patients undergoing surgery for prostate cancer, bladder cancer, or benign prostate hyperplasia. We had 322 patients, of which about 90% were Caucasians, mirrors the composition of the different ethnic groups here in greater Minneapolis. We had data on smoking status, and again, surgical procedures, prostatectomies or bladder cancer patients in which their prostate was normally benign prostate was removed. And we had benign prostatic hyperplasia patients. We had data on prostate-specific antigen levels, and also Gleason Score. Gleason Score, for those who are not familiar with this, it's a two-score test system which characterizes the morphology of the prostate glands and the degree of aggressiveness and the potential outcome of the disease, and also cancer stage. So we looked at a number of different biomarkers, including hair, which I'll discuss first, and then some DNA addicts. And we also looked at some oxidative stress markers. These addicts are formed from acrolein, which is an oxidative stress marker, which derived from lipid peroxides, and also some of these 4-hydroxy-2-alkenyls, which are longer chain derivatives of acrolein, which we uncovered during some of our DNA addict analyses. So the idea was, we know we can measure FIP in hair. We have some data that some patients do have damaged DNA because of FIP. And so we wanted to know whether elevated levels of FIP in hair, whether they correlate with aggressive prostate cancer. And Doug Cian actually insisted that I put this lower bullet line in my talks. It's maybe a little bit of an overstatement, but the idea is if we can validate whether cooked meat is a risk factor for prostate cancer, and FIP, which we think is a prostate carcinogen, we could monitor people and give them advice about changing diets and hopefully decrease risks for prostate cancer later in life. Because most 25-year-olds, they don't really care about prostate cancer. And it's only when you get to my age that you start worrying about this kind of stuff. I mean, I ate a lot of well-done burgers when I was younger. Okay, so the first thing we did was we had a self-administered food frequency questionnaire, which we adapted from the literature. And we asked the patients how many times per week do they eat meat, poultry, and fish. And you can see that we have considerable variations. We have a number of subjects with high levels of FIP in their hair, but also very low levels. And if you look at the correlations, I mean, they're not very good. The correlation between FIP in hair and the frequency of consumption, well, there's no correlation at all, whether we're normalizing for melanin, all right? So there are either two explanations for that. Either our analytical assay is really bad, which I don't think so, because we've shown the reproducibility of this, and coefficients of variation are less than 25% over subjects when we assay them over a 6-month period. Or else people have different perspectives of what well-done cooked meat is. And as I mentioned to you, the levels of FIP that are formed in these different meat staples, poultry, they can vary by over 100-fold. And other groups who are epidemiologists that have developed more sophisticated FFQs than ours, with diaries and repeated questions to the patients, they also concluded that FIP biomarkers, meaning urinary metabolites, poorly correlate with the FFQ, and the amount of FIP intake was grossly underestimated from the FFQ. So I'm a strong advocate of the FIP hair biomarker and assessing intake. So going back to this cohort here at the University of Minnesota, the first way we looked at the data is we asked how much FIP is in hair in subjects who had benign prostate hyperplasia, those who had bladder cancer in which their prostates were non-malignant. And you can see there's actually no differences in the levels of the addicts between these two subgroups. In contrast, the patients who had prostate cancer were significantly higher than either the bladder or benign patients. This is the first data set that correlates FIP in hair with prostate cancer, or well-done cooked meat containing FIP with prostate cancer. Then we initially did look at PSA levels as a continuous variable. Although there was a correlation with increasing concentrations, it did plateau off and somewhat decrease at the highest amount of well-done cooked meats consumption. And so we dichotomized this variable to a PSA level of 4. So generally, this is a threshold level. Levels lower than 4 are not a red flag, but if a man goes to get his blood tested and he has a PSA level that's 4 or above, that's generally the time when he has to go to a urologist to get an exam, because there is an increased risk with PSA levels above 4, although I must confide that this is a controversial biomarker and there's a lot of false positives and false negatives, but elevated levels of FIP were seen with the moderate and aggressive PSA scores of 4 and above. We also grouped patients with who had benign hyperplasia or Gleason score 6, which is the lowest Gleason score in which patients are monitored. There's no surgery, there's no treatments, but those with Gleason scores of 7 and above, there are various types of treatments. And so this is these cutoff scores that the oncologists suggested that we use. And you can see that the levels of FIP, again, they are significantly higher in those patients with Gleason scores of 7 and above compared to benign patients or patients with Gleason score 6. So this is the first biomarker to link well-done cooked meat with aggressive prostate cancer risk, and does support some of the epidemiology paradigms on the role of well-done cooked meat and prostate cancer. So we have collected DNA from these patients undergoing prostate cancer surgery, and we looked for DNA addicts not only of FIP, but several other carcinogens present in cooked meats, including methyl IQX and carboline, the polycyclic aromatic hydrocarbon benzopyrene, which based upon immunoassays, there's supposed to be tons of this in prostate tissue. We haven't been able to find it by specific mass spectrometry methods. And we also looked at the environmental pollutant aminobiphenol, which is a human carcinogen. We have only seen DNA addicts of FIP thus far. We've gone through about 150 patients. And this is some of our earlier data. We had collected tissues from both the peripheral zone and the transition zone, and seeing we do have similar levels of addicts in both zones, although most prostate cancers originate from the peripheral zone. This is a histogram of our cohort, the levels of FIP in hair and the number of subjects. And what I want to point out is that this data is very skewed. Because of this and my knowledge about levels of FIP in hair, my conclusion at the time of surgery, most of these patients actually had low levels of FIP in their diet at the time of surgery. For the cohort, positive control cohort at the University of Hawaii, where we fed them burgers with 454 nanograms per day, which is the estimated average intake of FIP in the United States for young adults, the levels of FIP in hair, the U of Hawaii cohort's right there. So you can see that the levels of FIP in hair, they're really quite low in this cohort. And we have seen about 10% of the patients do have FIP addicts in their prostate at the time of surgery. We have no idea about the rate of repair of this addict in prostate or when the last time was they ate well-done cooked meat. We also looked at these propanol addicts derived from Alkaline, and we have much higher levels of addicts, more frequently detected, but these addicts were not associated with the PC pathology biomarkers that we had seen for FIP, interestingly. And again, I want to emphasize that we have a small cohort, a small percentage of African Americans, and can't draw many conclusions about African Americans from this study. We did, however, look at the levels of FIP in hair of African Americans versus Caucasians, and we reported either as per gram of weight of hair, and actually the levels of FIP are about almost 3-4 times higher than mean levels than Caucasians. But if you normalize it per melanin, and again, African Americans have much more melanin in their hair than the Caucasians, that difference narrows down. And with this few number of subjects, it's not statistically significant. We need to have a larger cohort of African Americans because I believe, or I'm hoping to show that indeed, they still have higher levels of FIP in their hair. There's just not enough numbers here to support it with statistics. What we did is we took the number of patients who were positive for FIP DNA addicts, which is, and also we had data on hair measurements. And what you can see is actually we get we're having an association. It's close to significance. And anecdotally, the two patients with the highest amounts of FIP DNA addicts were African Americans. The data is a little bit less stronger, the association when we normalized for melanin. And I believe it's in part because our cohort, it's aged men, and they have less melanin in their hair. And so when you normalize for melanin, for those individuals that got grayish white hair like me, or maybe Pete, well, Pete's darker than me, but it may be overinflating that ratio. So the normalization for melanin may not be a perfect method, but there is an association here. We need more subjects, and I think we need another cohort. So all the work I've reported thus far is it's targeted. We had ideas, we had thoughts about specific chemicals and exposures, and we tried to analyze for these endpoints and either to prove or refute our hypothesis. We have developed an untargeted method to screen for DNA addicts. All right, there's probably a lot of DNA damage and things that we don't even know that are present in prostate, for that matter. Steve, probably lung and many other organs as well. And so we developed an untargeted method called wide SIM, wide selected ion monitoring, MS to the second, tandem mass spec, where we take a small packet of ions and we fragment everything in there. Okay. And the way that the mass spectrometer works and these addicts undergo fragmentation at the MS to the second stage is they will lose deoxyribose. So we can screen for any putative DNA addict where we know, I can say, well, maybe this addict or the mass weight 490 is present or 570 is present. And if we can see their fragmentation product, which is the loss of deoxyribose, and that peaked co-elutes, that's strong evidence for a putative DNA addict. Now, we've done this and we published this work in 2017, but it's a manual, it's a very labor-intensive process. Since that, our collaborator, our bioinformaticist, Scott Walmsley, has come on board, work with us, and he developed this very elegant, beautiful algorithm that automatically can screen for putative DNA addicts. We give him our raw data files, and he crunches through the numbers and can spit out a number of putative addicts that we can go back and we can do further characterization by mass spectrometry to try to characterize them. And his publication was out in Analytical Chemistry last year. So using his algorithm, we looked at untargeted DNA addicts in prostate cancer patients with either high or low Gleason score. And this is called the volcano plot. And we have like two types of fractions of DNA addicts. We call a polar lipid peroxidation fraction, and then a bulky aromatic fraction. And one of the challenges, there are many putative lesions or addicts, we don't know what they are. This one that I've exaggerated size in green here, however, is statistically significant, and we have identified it as a DNA addict of formaldehyde. It is higher in prostate patients with high Gleason score, more aggressive cancer than those in low. We know there's more inflammation, oxidative stress, and metabolism going on. And this addict is statistically significant. It's also a variable important trajectory. It's significantly higher in high Gleason score patients. And with our untargeted addictomics approach, also, we identified other putative 4-hydroxy-2-alkenyl DG addicts, which are derived from lipid peroxidation products. Although it was a little bit confusing at first because there are several different isomeric forms, and we believe that they have ring opened to form these stable hemiacetals. The same type of chemistry occurs with glutathione conjugates of 4-hydroxynonenal. And what we've seen is that from all of these, the subset of patients, we had 6 high Gleason scores and 6 low Gleason score patients. The high Gleason score patients have higher levels of these addicts. Whether they're contributing to cancer or not, we don't know yet, but These are oxidative stress markers, and they are elevated in high Gleason score patients. So we're quite intrigued by this. Well, I just said that they're elevated in high Gleason score patients. So then we've gone to look at studies of PhIP in prostate cancer cell lines. LN-cap cells, I want to emphasize they are a cancer cell line. The panel and activities of enzymes involved in metabolism of PhIP and DNA repair of lesions are most likely very different from those that are expressed in normal prostate epithelial cells. We looked at the parent compounds, and we also looked at the synthetic genotoxic metabolites for toxicity. The parent compounds did nothing. In contrast, the genotoxic anhydroxy metabolites, particularly for PhIP, showed strong toxicity. This means that the enzymes involved in the first round of bioactivation, cytochrome P450s, are poorly expressed in the cell line, but the synthetic hydroxylamines are genotoxic. We've seen that levels of PhIP addicts are much higher than those of these other prevalent heterocyclic amines, and we get a nice dose response down to levels approaching human exposure. So PhIP is a potent DNA damaging agent in this cancerous prostate cell line. We've looked at different enzymes involved in phase 2 bioactivation, and it turns out that N-acetyltransferases are the key enzyme involved in bioactivation in the cell line. We can't detect NAT2, so I believe it's probably NAT1 that's catalyzing it. So this is our working hypothesis. We believe that most of the PhIP is metabolized to the hydroxylamine in the liver. It goes through systemic circulation to the prostate, where it can undergo further bioactivation by phase 2 enzymes, or possibly detoxification through glucuronidyltransferases or glutathione transferases as detox mechanisms. For future work, we're very interested in African Americans in health disparity. They have almost double the incidence in mortality of prostate cancer compared to Caucasians. We recently were awarded a catchment award here from the Masonic Cancer Center. Thank you, Doug. And we're working with Clarence Jones, who is a health advocate of the Human Partnership, and we are recruiting African American men and to further generate data on their exposure to this carcinogen via hair. We know nothing about the metabolism of this compound in African American men either. There could be some health disparities, particularly with some of the phase 2 enzymes like glucuronidyltransferases. So also, you know, I don't want people going home today and say, Jesus, I can only have bread and water. I can't eat anything, all right? But that's not the message I want to give. But just don't eat your meat burnt. And there are plenty of beneficial foods that we eat every day, from red, yellow peppers, tomatoes, blueberries, raspberries, broccoli, which quite frankly I do hate, but they're potent inducers of glutathione transferases, enzymes that inactivate carcinogens. Fortunately for me, I love red wine, particularly Bordeaux, and there are some chemoprotective agents in red wine. You just got to be a little bit careful because unfortunately, ethanol at high levels of ingestion, it's carcinogen, so that's a dual-edged sword. Also, roasted garlic cloves are chemoprotective. And perhaps the strongest data in the literature from epidemiology studies is roasted ground coffee. It's highly protective against colorectal cancer. And there's also data for prostate cancer. So, you know, drink up and eat your vegetables. So future research, we'd like to further our studies on PhIP in normal prostate cells from patients undergoing cystoprostectomy to understand how PhIP may be inducing oxidative stress and cell homeostasis. We want to understand in healthy normal cells the enzymes, panel of enzymes involved in bioactivation and detoxification. And also if PhIP well, we know it does bind to the androgen receptor in vitro, and whether it has any impact on the anti-androgens that are being used to treat prostate cancer and using healthy normal epithelial cells. And we believe that our PhIP biomarker can certainly be used more in clinical translation. We'd like to understand whether this biomarker and the associations we've seen here at the University of Minnesota, can we repeat this in other cohorts and see whether there is an association of this biomarker with high-risk prostate cancer patients. And again, there are health disparities in whether the correlation of PhIP and hair might be associated with higher levels in, say, African Americans than white men. And cooked meat is thought to be a tumor promoter. So also the hair biomarker FIP, maybe it can be used in assessing PC outcomes, prostate cancer outcomes, and reoccurrence. So the key takeaways, again, don't eat your meat too well done. Man has been eating well done cooked meats ever since the advent of fire close to 1 million years ago. I think at that time, men were more concerned about being killed by a saber-toothed tiger than they were about developing colon or prostate cancer. But times have changed, and I'm not trying to convince people to become vegans. I still enjoy a steak, burgers. I won't eat them well done. I'll try. I know how these things form. I can look at a burger and I probably tell you within 10-20% how many ppb of FIP you've got in there. So just cook it medium to medium rare. Okay, that's it. All right. So the FIP hair biomarker in our anecdomic data does support the paradigms, the epidemiology paradigms of well done cooked meat and aggressive prostate cancer risk. We know that FIP certainly undergoes bioactivation in humans. We know that glucuronidase transferases are important enzymes in detoxification. Public health institutes, i.e. including IARC, have been using some of our data in formulating either regulatory or public health recommendations for healthy diets, which it's nice to see. And, changing the public's perception about meat and a healthy diet, it's a long-term goal. Lastly, let me make some acknowledgments to my current and former lab members. Majda's work on the prostate's been really beautiful. BH, unfortunately, who's left us for bigger and better things, as has Jingshu, who developed the addictomics. Scott and his algorithm has been key for our work, and our continuous collaborations with Peter and his acumen for mass spectrometry are greatly appreciated. I'd like to thank Chris Wade and Perry Murigan and Joe Koopmeiners for the statistics, our collaborations with Loic Marshall at University of Hawaii, and the patients who have just been incredible, our funding. And I'd certainly like to gratefully acknowledge the support of the Minnesota Masonic Charities and their creation of the Sharing Cancer Causation for me. I've been truly blessed, and thank you very much for your attention.
Great, thank you. The first question comes from our virtual audience, from Sylvia Balbo. How easy and how accurate is the measurement of carcinogens in the meat products you are testing? In other words, what is the variability of the dose test?
Well, measuring these do I have to repeat this? You repeated this, so I'm
No,
good.
No, you are good.
All right. So we can measure these chemicals in cooked meats fairly accurately or reproducibly. You have stable isotopically labeled internal standards. And so our reproducibilities are within 10%. So that's not an issue. But I would say that it is possible that we are even underestimating the amount of these carcinogens in cooked meats. I don't know if it's by 10%, 50%, or 100%. At high temperatures, some of them will react with carbohydrates and proteins, some of which get digested in the stomach under acidic pH conditions to liberate the carcinogen. So I would say that the amounts that we're measuring are at the lower estimate of what the daily intake is. Now, the levels of these compounds in cooked meats, as I mentioned, they can vary by over 100-fold how you're cooking your meat. And so that's a variability that, I can't control. We've measured things in poultry at 500 parts per billion. Well, we've measured them also down at like 2 parts per billion, depending on how well done the cooked meat or chicken is. Sylvia, I hope that answered your question. I'm not sure.
Thanks, Rob. From all of the tumor DNA sequencing studies that have been performed by TCGA and other consortia, is it known what mutational signatures arise from these DNA adducts from from FIP. So your hypothesis is that the addicts, when repaired, can give rise to mutations that give
Yeah.
rise to the cancer. So do we know those mutational signatures and which ones are related to these things that you're measuring?
That's a good question, and it's a question that I hate to hear because it's a tough question for me to answer because most of the most cancer-causing agents do not provide characteristic fingerprint mutational signatures. The only two in the literature that are really clear cut are aflatoxin in liver cancer, and aristolochic acid in renal cancer and bladder cancer. The types of mutational signatures that FIP does induce are similar to some of those of oxidative stress mechanisms. And so it's going to be really difficult to pinpoint and say unequivocally based upon mutational signatures that, all these patients are because of got cancer because of FIP, and those are because of I don't know, some other exposure or something. So that's going to be challenging. I feel that what will strengthen the associations is if we can get better estimates on intake and exposure, and if we can ultimately look at certain types of metabolic genotypes or phenotypes. So for instance, some of the glucuronidacetyltransferases, the UGT1A1 family, efficiently detoxify this carcinogen, or the glutathione transferase 1A1 or 1A2 forms efficiently detoxify. If we see that individuals have high levels of exposure of FIP in their hair or eat a lot of well-done meat and they have elevated risk, say, of prostate cancer, I mean, that will strengthen the association. It will continue to help support the paradigm. But the mutational signatures, which are very important, it's not specific. And so,
Yeah, so many of them are still labeled as unknown.
right.
It would be great to be able to pin.
Yeah,
pin a label.
and I must confide that, I mean, FIP is, the mutational signature has been reported in I think it's a white blood cell line, and the panel of enzymes in there, I mean, may be such that we're not even getting efficient enough bioactivation. There are more studies that need to be done on mutational signatures, and it's something that if we can get funded, we hope to do with collaborators at Jerry Zavadil at IARC to better associate that. But it's an unknown. It's an unknown right now.
And just one quick idea. You got to create a phone app so you can take a picture of your meat and know within how many parts per billion how much FIP is in it.
Maybe I can patent it. I don't know. But when you've been doing this stuff for 30 years, I mean, it's
Rob, just the latest fad in cancer risk evaluation is cell-free DNA. Just identifying cell-free DNA as a screening pattern. I think President Biden made this a big part of his moonshot.
I'm sorry, what
Cell-free DNA, just DNA and plasma.
Oh.
So have you looked at assays to try to link the cell-free DNA? Because as I understand the assay right now, they're just looking for mutational patterns.
Yeah.
Natera and Signatera has an assay that looks at mutation, but
Doug, I'm not sure you're gonna like my answer.
okay.
Well, maybe if we get one of these really super expensive FTIR mass specs that are about $10 million and much more sensitive, we may be able to detect those. I don't know, but the problem is those assays with the free circulating DNA. The problem is that they're doing PCR, they're amplifying things to get signal. We can't do that. We can only take what the DNA actual amount is, and then we have to break it down enzymatically. And the problem is that the amount of DNA that's circulating, I mean, it's very low. I mean, it's well below a microgram, I think, you get out of these types of systems. And it's just, we're not at that level of sensitivity.
Unrelated question. So when your dog was a puppy, this got your attention. Have you looked at any children? Children don't tend not to smoke, but children can eat a lot of meat.
No, I mean, that's a great question. No, I have not. And undoubtedly, as people age, their diets are going to change too over time. And Yeah, no, that's a great question. I have not. And, I don't know if McDonald's or Burger King would be happy about us doing that, but yeah. Um,
Great, we'll take our last question.
Rob, that was great. I had two questions. One is on the nitroso, right? Have you measured when you look at the NAT, does it inactivate NAT1? Have you detected inactivation? Because those nitrosos are potent inactivators in NAT1. I was just curious
Right.
if that one is, and have you seen a decrease in NAT1?
I remember your paper with Pat on that. That was beautiful, beautiful work that you guys did. We have not looked at its ability to actually deactivating the enzyme because that sulfur is and cysteine is in a critical active site. No, we haven't, but I don't know how many turnovers you would have before, but I would suspect that some of the chemistry that you reported it was on nitrosofluorine, right?
Right.
I'm sure it's going to be similar, but whether nitrosofip would be more potent, less potent, I don't know.
Oh, yeah. I was just curious if that then skews because you're inactivating the NAT and elevating. And the second question is, where does the FIP? Do you know where the FIP comes from? And in the molecular, in other words, does it come from tryptamine? Where is it coming from? And the question is, in these meat substitutes now that are like Beyond Meat and all, is it less likely that you would get FIP from those substitutes? So you'd have a reason.
Yeah, I've often wondered about that, about these artificial meats, or Donald Trump would say fake meat, I guess.
Mm-hmm.
But first of all, the FIP, maybe I should put a slide back in. I just thought I had too many slides. So first of all, creatinine is a critical component precursor for the formation of FIP. That's coming, the N-methyl aminoimidazole moieties from creatinine. Okay, the FIP, it's coming from the other precursors are fragmented hexoses and losing my brain. It's not phenyl. I should know this. It's not phenylalanine. Well, there's multiple amino acids that can contribute to it, but you're getting fragmented hexoses that are combining with creatinine and degradation products of several amino acids that combine to form it. These model studies have been done like 30 years ago, which I should remember this, but so the precursors are well known, and next time I'll put a slide back in, Rick. But but as far as these Beyond Meat or the Impossible, whatever, I thought about that. I've eaten some of those products because I was curious. Actually, I think the sausages are better than the burgers, but I don't know whether they form or not because I don't know how much, if any, creatinine is in there. All right. But of course, you cook this stuff at high temperature, it's proteinaceous. There could be other heterocyclic type amines that are producing that. Nobody's looked at that, to my knowledge.
All right, great talk. Everybody say thank you one more time to Dr. Turesky.